So, the B cell receptor, or BCR, is composed of a monomer of an antibody, no matter what class of antibody. And that means whether it's an IgM, IgA, IgG, IgE, B cell, it is still a monomer. So even though you, for example, will secrete an IgM that's a pentamer, so you have 5 monomers attached together by the J chain, that B cell, before it gets stimulated to differentiate to plasma cells, the receptor will be an IgM, but it'll be a monomer. Same with IgA, it will be a monomer. Okay? And the B cell receptor also is composed of a couple other components, Ig beta and Ig alpha. As you can see in this little cartoon, the IgM is attached into the plasma membrane, and there's very little that goes into the cytoplasm. So there's really not much there to allow the B cell receptor, the monomer, to stimulate or communicate with the cell to let it know it's been engaged. However, there's 2 other proteins, the Ig beta and Ig alpha, that are also associated with the monomer, and this together is the B cell receptor, and it has a longer tail that has a tyrosine-based activation motive, or ITAM, the immunoreceptor, and you can see that on the cartoon. And so that's actually what stimulates the signal to the cell. So basically, the B cell receptor and the co-receptor, which was the cooperating B cell activation. So the B cells become activated when the receptors are cross-linked by antigens. So you can see it's just not simply an antigen binding to the B cell receptor. It's multiple receptors on the surface of the B cell that are being engaged simultaneously. They're getting cross-linked. And that sends a signal to the cell. Now there's a co-receptor that is also involved. It amplifies that signal, and that co-receptor is CR2, and it is able to recognize C3D. And C3D is a breakdown product of C3b. That remains bound to the surface of an antigen pathogen. So this B cell co-receptor, which has CR2 along with CD19 and CD81, as you can see in that second panel, together with antigen engaging the monomer of the B cell receptor, and the recognition of not only the antigen but the complement degradation product C3D together is the optimal way to send that first signal to the nucleus for activation to occur. Now, there are 2 major types of antigens that can activate a B cell. They're the T-independent, or TI antigens, and the T-dependent antigens. Now, the T-independent TI antigens, they are called that way because they do not require T cells. So an athymic individual who has no T cells can still develop an antibody response against certain antigens. The responses are predominantly IgM, they're low affinity, they have no memory. So this is a T-independent antibody response. IgM, low affinity, no memory. And you can break those down into 2 different T-independent antigens, the TI1 antigens and TI2. The TI1s are also known as mitogens, or polyclonal activators. That means that at high concentrations, they can stimulate B cells to be activated. But at low, irrespective of the specificity of that B cell. Okay, so even if it's not, that B cell receptor doesn't recognize that mitogen, that TI1 antigen, it can get stimulated as long as there's high concentrations. On the other hand, at low concentrations, they can stimulate a B cell only if that B cell recognizes that antigen. So if a B cell is specific for that TI1 antigen and it engages the B cell receptor, then it has the capacity to stimulate that cell even though it's not getting T cell help. The TI2 antigens are repetitive epitopes. They have lots of, they're multivalent, they have lots of epitopes. Example is carbohydrates in the capsule of a bacteria, or the flagella, or some sort of repetitive unit type thing that engages. And there's a specific B cell subpopulation that respond to those, and those are the B1 cells. Respond to those. Okay. Now the T-dependent antigens requires T cells. So T-independent antigens, you don't need T cells. T-dependent, you have to have T cells. So protein antigens, typical protein antigens, most really good antigens will be a T-dependent antigen. They will trigger memory. You will also have the potential of isotype switching, so you could change the antibody class from IgM to IgG. You can have increases in affinity through a process called somatic hypermutation. So that's fine-tuning the antibody specificity, making it more specific for that particular antigen. So let's again talk more about the T-independent antigens. These T-independent antigens do not require T cell co-stimulation. The TI1 antigens, a good example is lipopolysaccharide, or LPS. It's an endotoxin of E. coli and other gram-negative bacteria. The O region binds the BCR. That's the O region is the carbohydrate portion of it. Binds at the B cell receptor, and the lipid A binds to a toll-like receptor, TLR4, on the surface, and that combination helps stimulate that B cell to become activated. The TI2 antigens are typically polysaccharide antigens. These are examples. For example, Streptococcus pneumoniae has these polysaccharide capsule that coats it, prevents it from being easily phagocytized unless there's an antibody response against it. And as polysaccharide antigens have repeated epitopes that cross-link multiple BCRs. Now the big thing about the T-independent antibody responses is they're relatively rapid. They can happen within days, 2 to 3 days. They result in low affinity IgM, and they're short-lived plasma cells. So an example is the T-independent B cell active TI1 antigens. You've got LPS as a TI1 antigen. It can be recognized, the carbohydrate portion can be recognized by the B cell receptor, and portions of it will also stimulate the toll-like receptor 4, and that results in the IgM antibody specific for lipopolysaccharide. Now, at high concentrations of LPS, you don't need that binding to the B cell receptor. It can interact directly with TLR4, and that'll stimulate the response by itself. But at low concentration, it'll stimulate antigen-specific B cells. T-independent B cell activation, the TI2 antigens. Again, a good example is the polysaccharide capsule that you see on the surface of bacteria to protect them from getting killed, and you need repetitive epitopes, so a B cell will bind to it through multiple B cell receptors, and that gets engaged, and that'll stimulate that B cell to become activated. So, to kind of summarize, the T-dependent and the TI responses, to get that straight, is the T-dependent antigens as a soluble protein. It's a humoral response would be include isotype switching, affinity maturation, immunologic memory. The T-dependent antigens don't typically are polyclonal activators. The TI antigens, there's type 1 and type 2. Type 1, bacterial cell wall component, like LPS. You don't get isotype switching. You don't get affinity maturation. You don't get memory. However, you can, at high doses, get polyclonal activation with the type 1 TI antigens. TI2 antigens, you can get limited isotype switching, but it's not characteristic. You get you won't get affinity maturation, immunologic memory, and it will not activate B cells through polyclonal activation. Key things here is the TI antigens generate IgM responses prior to the T-dependent responses. So again, it only takes 2 to 3 days to get a T-independent response. However, it takes 2 to 3 days to take a T-independent antigen response. However, it takes longer to get the T-dependent responses. So it's thought that the T-independent antigen responses, the antibody responses, help to provide early defenses against infection. And so while the T-dependent responses are more specific and have greater functional activity and larger. So the idea is these B cells get activated earlier, provide some early level of defense while the body is mounting a more effective response. And so it might control the infection and keep it from getting out of hand before the really good adaptive T-dependent response is generated. So thymus-dependent TD antigens. So it requires T cells. Requires specifically CD4 T cells. The CD4 T cell provides the costimulatory signal that promotes B cell proliferation, the generation of higher affinity B cell receptors. The T helper cell also can facilitate or signal to the cell isotype switching. It helps determine the signals sent, determines which isotype to switch to. So it changes from an IgM or to an IgG, an IgA, or IgE, which ultimately will be what that B cell can produce when it differentiates the plasma cell. The T helper cell also participates in memory formation. So that means that the next time you see that antigen, it'll generate a better increased affinity, higher response, and of a different isotype than IgM. The downside of this type of response, it takes a bit of time to generate. And so it's a slower antibody response. It takes 5 to 10 days. So you can see where the T-dependent antigen responses helps bridge that time, so you can get an antibody response showing up within a couple days. That's low affinity, but it still is could help control the infection and keep it from getting out of hand, while the T-dependent responses develop a few days later. So how does B cell activation occur? Well, the B cell itself can act as an antigen presenting cell, and the B cell is really good at presenting antigen that it's specific for. So basically, as you can see in this cartoon, a microbial protein antigen is binding to the B cell receptor. It gets endocytosed, and so that microbial antigen gets processed, it gets degraded into peptides, and during those steps, it gets loaded into the MHC class 2 molecule. And when that occurs, now an effector CD4 positive T cell can now recognize that. Okay, so you have a class 2 with a peptide. If the T cell is specific for it, it can recognize that, and in doing that, it'll generate that second signal that helps stimulate the B cell to proliferate and go through somatic hypermutation, more higher affinity, isotype switching, and differentiation eventually into plasma cells. Now, T-dependent responses develop in secondary lymphoid tissues. So secondary lymphoid tissues are ideal for getting B and T cells and other antigen-presenting cells together. It forms that microenvironment, the structure for those interactions. And here's a cartoon where you got naive CD4 positive T cells are activated by antigens presented by dendritic cells in the lymph nodes. So a T cell wanders in from the circulation, crosses into the lymph node through the high endothelial venules, the HEV. When it gets into there, it'll go around looking for antigen-presenting cells, and if it sees it, the T cells can get activated. So you now have activated T cells there in the lymph node. Also, naive B cells also migrate into the lymph nodes. If they have been activated, if they have seen antigen that they're specific for before or while they're in the second lymphatics or the lymph node, and they bind to it, then they're in the perfect place to present antigen. They pick it up, present it, class 2, and CD4 cells that have also been activated will then recognize that, and you'll form these conjugates between the antigen-activated B cells and the T helper cells. And then, so when they form those conjugates, that is when you can get activation of the B cell. So again, the B cells prevent specific antigen to T helper cells, and then those T helper cells recognize that and deliver the second signal. The second signal here, as you can see in the panel 2, is CD40L on the T helper cell and CD40 on the B cell. You can also get cytokines produced by the T helper cell that could help activate and influence differentiation of the B cell. So again, as you see in the first panel, the B cell recognizes that antigen, brings it in, degrades it, presents it on the surface with class 2. A T helper cell that's already been activated recognizes that, then delivers a second signal, the CD40L and the CD40, and cytokines that will deliver the signals for B cell activation. And so those cells can go on based on those signals to switch, increase affinity through somatic hypermutation, differentiation of plasma cells, memory cells, whatever's necessary. So this all happens in the lymph nodes. The B cell proliferation, differentiation, somatic hypermutation occurs in germinal centers. And so when you get this activation occurring, you have certain cells. You get the B cells and the T cells together, along with another cell type called follicular dendritic cells. These follicular dendritic cells capture immune complexes with antigen on the surface and present it to the B cell. So What happens here is these follicular dendritic cells, T cells, the B cells are now proliferating, they're differentiating, they're fine-tuning the specificity of the antibody through somatic hypermutation, they're differentiating in terms of isotype switching, etc. And you have these dense masses that are called germinal centers, and it's partially can contribute to things like increasing the size of some lymph nodes because you've got active immunity occurring there. Now, the T cell can also deliver a couple signals that will influence, help promote plasma cell differentiation. And the two major ones we're going to talk about is APRIL and BLYS, or BAF is another name for it, B-lyse and BAF. And these can be receptors on the surface or released as a soluble protein, and they'll interact with BR3, the B-lyse receptor, TACI, BCMA. There's also a couple other molecules on the surface that can participate in this. So APRIL is a cytokine proliferating inducing ligand, B-lyse or B cell lymphocyte stimulator, BAF is a B cell activating factor, TACI is a membrane activated calcium modulator cyclophilin ligand interactor. Boy, that's a mouthful. BCMA is a B cell maturation antigen. Okay, so you have this interaction here, and that actually helps stimulate plasma cell differentiation. Now, there's actually a therapy that looks like it might be pretty good with certain situations. It's atacicept. It's a fusion protein of the TACI receptor. And basically, what we mean by fusion protein is typically you can take the receptor, you fuse it to the Fc portion of an immunoglobulin molecule. So you now make it soluble. It lasts longer in the circulation because of that fusion. If you don't have the Fc portion on it, it gets cleared relatively quickly. And so basically what this does is it will bind to those cytokines, the APRIL and B-lyse cytokines. And so you can see TACI is one of the receptors that normally have specificity for both of those cytokines. And so the idea, if you give this to a patient, this soluble fusion protein to a patient, it'll bind up these cytokines, these signals, and block these signals, and not interfere with B cell differentiation. So it blocks B cell differentiation into plasma cells by interfering with the binding of soluble B-lyse and APRIL. It has been shown that I think it's gone through phase 3 trials now. It shows efficacious against systemic lupus erythematosus, or SLE, or lupus, which is an autoimmune disease where B cell activation is a major component of it, and it causes a lot of soluble immune complexes and attacking cells, as well as depositing those immune complexes in kidneys, skin, lung, etc. There was a trial for multiple sclerosis that looked promising early on, but I gather it failed phase 2 trials. And actually in patients, multiple sclerosis patients, they were found to have increased relapses than those that were given placebo. So they ended the study early because there was obvious concern. Now you could have genetic defects that can interfere with B cell development. And a good example is Bruton agammaglobulinemia, or X-linked agammaglobulinemia, or XLA. You'll see all those terms. And what it is a mutation in Bruton's kinase gene, or BTK, and it's important in B cell maturation. If you have a mutation in this, no B cell receptor is made. The BTK controls rearrangement of its IgM heavy chain during B cell development, and so if you don't get B cell IgM formation, the heavy chain formation, you don't get the B cell fails to fully develop, and they're unable to respond to most antigens. These patients, they basically make little or no antibody, and they have a high rate of infections, particularly against bacteria, and mainly encapsulated bacteria. They could still fight off viral infections because you have a good T cell response, but the problem is that these patients may also be susceptible for repeated viral infections because the antibody response is important in preventing additional infections. Treatment of these patients is usually an IgG passive immunotherapy. So you give human gamma globulin to provide immunity against many of these bacteria. In kids, you may be concerned with certain agents such as respiratory syncytial virus, which is a very common and very problematic child who doesn't have a good antibody response, has this defect. And so you can actually give more specific antibodies that are specific for respiratory syncytial virus, for example. The other thing that's typically done is through prophylaxis, you may give these patients long-term antibiotic treatment to prevent any significant infections.